deciphering the Rhizosphere microbiome for disease-suppressive bacteria, I would first like to introduce the pathogen which this study is based upon, and the reason for this investigation, Rhizoctonia solani. Rhizoctonia solani is a soil-borne pathogen which attacks the roots of important agricultural crops such as sugar beet, potato, and rice, causing serious economical impacts, especially as seedlings. The current trend in plant disease suppression is to increase this disease suppressiveness by manipulating the microbiome around the rhizosphere. The rhizosphere is the area of soil adjacent to the roots that is directly affected by plant secretions. A suppressive soil is a soil which susceptible crops don't suffer economic losses despite the presence of the pathogen. The opposite, a conducive soil, is a soil which the plant progresses on, the disease progresses uninhibited. The aim of this study is to identify the bacteria and its genes associated with a soil suppressive to R. solani. A soil suppressive to R. solani was found in the Netherlands. The authors then prepared 5 soil treatments: suppressive, conducive soil amended with 10% suppressive soil, and soils heat treated at 50 and 80 degrees Celsius. As you can see by the graph, the amended soil had a partial transfer of suppressiveness, and the heat treated soils had partial or complete loss of suppressive functions, which proves that the suppressiveness is biological. A final treatment was prepared that was a suppressive soil inoculated with R. solani to identify any taxa that reacted to the presence of the pathogen. The authors then used 16S oligonucleotide microarray, also known as phylotype analysis, to identify the bacteria in each of these treatments. They then compared the differences between suppressive, conducive, the amended and the inoculated soils to find the bacteria associated with soil suppression and their abundance. Beta proteobacteria was the bacteria most associated with soil suppression, and they then focused their study on a group of beta proteobacteria called Pseudomonads, which has already been associated with the suppression of other fungal pathogens. A pseudomonad was then isolated from the suppressive soil and grown on a selective medium. They then used DNA fingerprinting to identify the similarities between the DNA and allowing them to group them into 10 haplotypes, that is, DNA similar groups. They then tested the suppressiveness of each bacteria against R. solani. The most effective bacteria then mutated, making it no longer function, and then allowing them to see the genes that actually functioned as a suppressive, helped it function suppressively. The authors of this paper achieved their aim, which was to identify the major families of bacteria associated with a suppressiveness of R. solani. Additionally, by focusing on a single group, they were able to identify the genes involved with this as well. However, they also conclude that because pseudomonads were not the only bacterial family associated with disease suppressiveness against R. solani, that it's more likely to be due to a consortium of bacteria. Thank you.